Okay, so the second part of the PowerPoints are going to be looking at the function of neurons and how they create a nervous impulse. And the first thing that we have to look at is the state of the neuron when it is at rest. So when a neuron is at rest, it's the state of its membrane is under a negative charge. So the resting membrane potential of the neuron is minus 70 millivolts. So this is the resting membrane potential of the neuron. The negative charges are coming from negatively charged proteins inside the neuron. Because proteins are large molecules, they cannot leave the cell, and so the negative charges are permanent fixtures of the neuron. On the other hand, ions found inside the neuron and outside of the neuron in the interstitial fluid also play a role in creating the nervous impulse. The outside of the neuron, the interstitial fluid, has a high concentration of sodium ions, making the area positively charged. On the other hand, the inside of the neuron has a high concentration of potassium ions. Potassium ions are not in high enough concentration to offset the negative charges of the proteins, but it does create a higher concentration of these potassium ions inside the cell than outside the cell. Now, if you remember from Biology 1406, how diffusion happens from an area of high concentration to low concentration, you could see that if sodium channels were to open, sodium will diffuse inside the cell following the concentration gradient. By the same token, if potassium gates were to open, potassium would diffuse out of the cell following a concentration gradient. So as long as gates are closed, the resting state of the neuron remains at minus 70 millivolts. Okay. So the resting membrane potential is due to the negative charges brought about by proteins inside the cell. But there's also a difference in the ion concentration, the inside of the neuron and the outside of the neuron. Remember, inside the neuron there is more potassium, outside the neuron there is more sodium. So that here you can see the negative charges of the neuron inside. Outside is positively charged because of the potassium. Not potassium, sorry, because of the sodium. Okay. So in the intercellular fluid has lower concentrations of sodium than extracellular fluid. Intercellular fluid has higher concentrations of potassium and negatively charged proteins than extracellular fluid. Okay. So this is showing you different types of channels. Here we have a potassium channel, and when the potassium channel opens, potassium leaves the cell following the concentration gradient. This is a sodium channel. When the sodium channel opens, sodium goes inside the cell following a concentration gradient. This right here is the sodium-potassium pump, which is going to spend energy bringing sodium, taking sodium out of the cell, sodium out, and bringing potassium into the cell. Okay, so as long as the membrane is at minus 70 millivolts, there is no nervous impulse being created. A nervous impulse is created when there is a reversal of the charges of the membrane. This is brought about by changes in ion concentration between the outside and the inside of the membrane. So that's what's going to cause changes in membrane potential. A change will be a signal. So in this case, the nervous impulse, the signal, is created by changing by changing the charges inside the neuron. There are two types of changes that happens in the neuron. There's the change that is brought about by the stimuli that arrives at the receptive regions of the neuron. So stimulus arriving at dendrites and the body of the neuron is going to open sodium channels. When sodium channels are opened, sodium flows inside the cell, bringing with it positive charges. A change in voltage follows, and that change in voltage is called the graded potential. An action potential, on the other hand, is a long-distance signal, a long-distance change of charges that happens along the axon of the neuron. So when ions move in and out of the cell, if ions move into the cell, let's say sodium ions move into the cell, making the membrane potential more positive, that is called depolarization. So a reduction in membrane potential towards zero, meaning the membrane potential changes from negative to more positive, is called depolarization. Depolarization could lead to a nervous impulse or action potential. So during the influx of sodium ions, there is a gain of positive charges. The gain of positive charges makes the membrane more positive. This event is called depolarization. So here's depolarization of the membrane. The membrane is at rest. Sodium ions enter the cell and the membrane becomes more positive or less negative, and that's depolarization. Hyperpolarization, on the other side, is an increase in the membrane potential away from zero, meaning the membrane becomes more negative than minus 70 millivolts. This happens when potassium ion gates open and potassium leaves the cell. This type of graded potential is going to reduce the chances of creating a nervous impulse. So if the stimulus, let me go back really quick, if the stimulus causes hyperpolarization, okay, so if the stimulus causes hyperpolarization by opening potassium channels and allowing potassium to leave the cell, an action potential or nervous impulse is less likely to be created. On the other hand, if the stimulus opens a sodium channel, okay, so the stimulus opens sodium channels, sodium now will go into the cell, making it more positive, and that increases the chances of creating a nervous impulse or an action potential. So during the outflux During the outflux of potassium ions, there is a loss of positive charges. This loss of positive charges makes the membrane more negatively charged. This event is called hyperpolarization. And here's hyperpolarization. The membrane is at rest, becomes more negative. That's hyperpolarization. So a graded potential is the reversal of charges that occurs due to the arrival of a stimulus. A stimulus is going to arrive at the receptive region of the cell, which is the dendrite and the body of the cell. Graded potentials are short-lived, localized changes in membrane potential. They can lead to depolarization or hyperpolarization of the membrane. So if this is a little neuron, when the stimulus arrives, and the stimulus can be something like touch, Could be a chemical, could be a change in temperature. The stimulus could open sodium channels, and then sodium is going to go into the cell. That will lead to depolarization. On the other hand, the stimulus could open potassium channels, and when that happens, potassium leaves the cell, making the cell more negative or hyperpolarizing the cell. This makes it less likely that an action potential will happen. So here's a stimulus arriving. The stimulus is a needle poking the skin, for example. This causes an opening of sodium channels, and a wave of sodium goes into the cell. Because positive charges repel each other, they're not going to remain hover next to each other. Moreover, there are negative charges surrounding them. So these positive charges will begin to travel away from the source, and the graded potential will be spread in this manner. So this influx of charges due to the stimulus is what we call the graded potential. And graded potential travels as the charges repel each other. So that as we move away from the source of the stimulus, the graded potential becomes less strong. It diminishes in magnitude. Graded potentials are short-lived signals, and the magnitude of the graded potential varies in proportion to the magnitude of the stimulus. So a strong stimulus will give you lots of a strong graded potential. A weak stimulus will give you a weak graded potential. And this is showing you how the graded potential is called graded because it's strong at the area where the stimulus is acting, and as you move away, it diminishes. So it's an important concept to know that graded potentials can be large or small depending on whether the stimulus was large or small, because action potentials are never that way. Action potentials are always the same regardless of the stimulus. So graded potentials begin at the receptive region of the neuron, which are the dendrites or the soma. Oh, come on. Okay. All right, so what we have is a neuron that is receiving a stimulus. And again, the stimulus can be something like a chemical that arrives at the neuron. Okay. The chemical can be a neurotransmitter that is being released by another neuron in the pathway. And the receptive regions of the neuron, which are found in the dendrites and the body of the neuron, has chemically gated channels that receive the chemical and open the sodium channels, causing an influx of sodium into the cell. So that would be an influx of positive charges into the cell. The positive charges begin to travel, and they get to the area of the cell called the axon hillock. At the axon hillock, there's a different kind of channel. There we have voltage-gated channels, meaning that these channels are going to open when there is a change of voltage in the cell. Voltage-gated channels are going to be found starting at the axon hillock and down the axon. These are the channels that are going to open only when there is a reversal of charges. So the stimulus creates a graded potential, which is a reversal of charges that travels to the axon hillock. And as more and more positive charges travel at the axon hillock, eventually there will be enough positive charges to bring the area from a minus 70 millivolts to minus 50 or 55 millivolts, And at that value, the voltage-gated channels open. So let's look at what happens. An action potential is a brief reversal of membrane potential with a total amplitude of about 100 millivolts. Okay. Notice that the action potential does not increase or decrease in magnitude over distance. It is always the same. It is not graded. So an action potential cannot be large or small. An action potential is always the same. An action potential travels the long distance from the axon hillock to the end of the neuron. So the stimulus will arrive at the receptive regions of the neuron, which are going to be the dendrites and the soma. The stimulus causes a graded potential, which opens sodium channels, allowing positive charges of the sodium to move into the cell. Those positive charges are what we call the graded potential. The graded potential travels to the axon hillock, and the potential of the axon hillock begins to change from minus 70 millivolts to minus 69 millivolts. As more and more charges arrive, the potential at the axon hillock continues to decrease, become more positive and positive. Minus 68 millivolts, minus 67 millivolts, and so on. When the potential reaches minus 50 or minus 55 millivolts, that's the value that we call the threshold. At that value, voltage-gated channels open. Sodium voltage-gated channels open very quickly, and a flood of sodium all of a sudden enters that area of the neuron. Keep in mind, the positive charges that arrive there arrive by the graded potential. All of a sudden, when the area reaches minus 50 millivolts, voltage-gated channels open, and the flood of sodium that arrives will be the beginnings of the action potential. Okay, so there is the influx of sodium, which is going to increase the membrane potential from, at this point, it is minus 50 millivolts, and the flood of sodium is going to increase it to plus 30 millivolts. It's going to make it very, very positive. At plus 30 millivolts, voltage-gated sodium channels are going to close. They're going to be inactivated, and by then, potassium channels have opened up, and potassium begins to leave the cell, taking positive charges out of the cell. So now the cell will begin to turn around and become more and more negative. So this is a repolarization that will lead to a hyperpolarization. So more and more positive charges leave the cell until the cell reaches minus 90 millivolts. At minus 90 millivolts, the voltage-gated potassium channels are going to close, and now the resting membrane potential is going to be restored. So remember, minus 90 millivolts is the hyperpolarization of the cell, or of the axon hillock of the cell. Okay, so let's go back to the positive charges that are depolarized in the axon hillock, creating the action potential. Those positive charges continue to travel down the neuron, and as the action potential begins to diminish at the axon hillock, as the cell begins to attain membrane resting potential again, the charges have traveled to the next portion of the axon, and an action potential will begin at that portion. When the action potential begins to diminish at that portion, by then positive charges will have traveled to the adjacent portion, and an action potential would begin at the adjacent portion, and so on and so forth. So action potentials begin to spread down the axon. This is a representation of the action potential. you definitely need to know this curve and the parts of the curve, what is happening at each step. So this is the membrane at rest. And by the way, this is happening at the axon hillock, and then it will continue to happen down the axon. This is what we call the action potential. So the cell is at rest, or the area is at rest, when a stimulus has arrived that brings in positive charges. And so the area becomes more and more positive until it hits the magic number threshold, minus 55 millivolts. At that value, voltage-gated sodium channels open, and there is a tremendous influx of sodium, which causes the cell to become very positive very quickly. This is called depolarization. When the membrane reaches plus 30 millivolts at that value, voltage-gated sodium channels close and voltage-gated potassium channels open, and there is an outflux of potassium from the cell, making the cell more and more negative. It reaches minus 90 millivolts. That's what we call hyperpolarization. And at that point, voltage-gated potassium channels close, and the cell now needs to restore the resting membrane potential. The sodium-potassium pump is going to accomplish that by kicking out sodium and bringing in potassium. So at the resting state, all the sodium and potassium gates are closed. Depolarization happens when the gradient potential brings in waves of sodium ions, which depolarize the area until it reaches threshold. At threshold value, there is an opening of sodium-gated voltage channels, which is going to depolarize the cell to plus 30 millivolts. So at threshold, which is minus 55 to minus 50, the permeability of the membrane to sodium increases, and there is a sodium influx into the cell, and the cell membrane becomes more positive. That is this part of the curve right here. This is due to sodium influx. Once the cell hits plus 30 millivolts, sodium voltage-gated channels are inactivated and potassium channel gates open, and potassium begins to exit the cell, taking with it positive charges and making the cell more and more negative. So this is the peak right here, plus 30 millivolts, when potassium channels open and the cell loses potassium, becoming more and more negative until it hits minus 90 millivolts. Okay, so this is hyperpolarization of the membrane when it reaches minus 90 millivolts. It's more negative than resting state. Now, at minus 90 millivolts, all voltage-gated channels are closed. The cell, however, has remained with high sodium and low potassium due to the depolarization stage. So the cell needs to go back to its resting state, which is high potassium, low sodium. It does that by using the sodium-potassium pump. So this part right here of the action potential that returns the membrane to threshold value, minus 70 millivolts, is accomplished by the sodium-potassium pump. So action potentials are what we call are all or none event. They either happen or they don't. Once they begin, they're always the same. There is no variation. Always the movement, the changes will be from minus 70 millivolts to plus 30 millivolts, from plus 30 millivolts to minus 90 millivolts. And this is showing you the permeability of the membrane to sodium and potassium. So again, I cannot emphasize enough that the action potentials are always the same. What is going to vary is going to be the frequency of the action potential. So at plus 30 millivolts, sodium channels close and potassium channels open. The outflux of potassium charges causes a rapid hyperpolarization to minus 90 millivolts. At minus 90 millivolts, potassium channels close. The cell is brought to resting membrane potential and ion balance by the sodium-potassium pump. Okay. Okay, so which are true of action potentials? They travel down the axon. They're always the same. They start at the axon hillock, and that's it. Okay, so once an AP, an action potential, is produced, the axon hillock, at the axon hillock, currents of these positive charges are going to affect adjacent areas, and the action potential always travels down the axon in one direction. So here's the action potential that is being created, and it's traveling to the next area. In this picture, the area in gray has just experienced an action potential, and it cannot experience another one until everything is brought back to resting state. The reason why this area cannot experience another action potential for a little while is because at that point, the potential of the membrane, sorry about that. Okay, sorry. Yeah, at this point right here, the potential of the membrane is not minus 70 millivolts. At this point, it could be plus 30 millivolts, depending on at what point of the action potential that area is in, or it could be minus 90 millivolts. So there's not an action potential that can happen at those values. So while this area is creating an action potential, the previous one that is finishing an action potential will not be at rest. And that's why arriving positive charges from a stimulus are not going to create an action potential in the area at that point. But this area is going through an action potential, which means that the next area to experience an action potential will be the adjacent area right here. And in this manner, action potentials always travel forward down the end of the neuron. Okay. All right, all action potentials are alike and are independent of the stimulus intensity. So how does the central nervous system interpret action potentials? Well, it's not based on whether they're large or small, they're based on the frequency. So a weak stimulus leads to less frequent action potentials. A strong stimulus gives us much more frequent action potentials. Okay. So what causes this action potential to move down the axon instead of moving back towards the body, or instead of going in both directions, is this absolute refractory period that the area enters as soon as it finishes or while it's going through an action potential. So when an area of the neuron is under a refractory period, so let's say this area in blue is under a refractory period, that means that when the action potential is being created right here, the positive charges could travel back, but they're not going to start an action potential in the place where they had just finished one. So in this example, this area is at rest. the area in gray is under absolute refractory period. So it is under minus 90 millivolts or plus 30 millivolts. Whereas the next portion is where the action potential is actually taking place. Positive charges arriving at the previous area are not going to initiate another action potential. There's not enough of them for that. But the positive charges arriving at areas that are down the axon could create an action potential. So that the point here is that action potentials always travel down the axon. There are two ways to move action potentials. Here, it could be by continuous propagation, which is what we've been looking at, in which action potentials are created at adjacent areas of the neuron until the action potential reaches the end of the neuron. So how fast the action potential travels down the axon depends on the diameter of the axon and the degree of myelination of the axon. The greater the diameter, the faster the action potential travels down the axon. The larger the axon, the faster the action potential will travel. Myelinated neurons, the myelination of the neuron is causing the action potential to travel very quickly because the action potential is going to jump from node of Rambier to node of Rambier to node of Rambier. The areas under myelination has no ion channels, so they cannot contribute to the action potential. Instead, the action potential travels in the little exposed areas between nodes of Rambier. And the myelination will cause the action potential to travel very quickly down the axon. Okay, so here's again saltatory propagation, which means jumping, means that action potentials only happen at exposed areas of the cell. So the action potential literally jumps from one place, from one adjacent place to the next adjacent place. Okay, so continuous propagation is slower, saltatory propagation is much faster. Okay. If you have heard of a condition called multiple sclerosis, this is a condition, an autoimmune condition, which destroys the myelin sheath of the patient. This is especially damaging to heavily myelinated neurons, like neurons of than somatic sensory and somatic motor neurons. So these are large neurons with heavy myelination, and they are the ones that are going to be damaged by multiple sclerosis. The myelination is damaged, and the action potentials will not travel as quickly down these neurons anymore. So again, we can classify neurons based on the diameter of the neuron, the degree of myelination, which gives us the speed of conduction. So group A fibers are the ones that are very large, heavily myelinated. These are going to be somatic sensory neurons or somatic motor neurons. I should say somatic there. Okay, so somatic sensory and somatic motor neurons are group A fibers, large and heavily myelinated. This is why somatic motor neurons are going to innervate a skeletal muscle. And so these will allow us to use the skeletal muscle very quickly if we need to. If it is a situation of life or death, we can immediately begin using the skeletal muscle, and skeletal muscle, the signals will travel very, very quickly in the body to tell the skeletal muscle to begin acting. By the same token, somatic sensory neurons are bringing information from the outside body, information via the eyes, via the hearing, which can be crucial information for survival. So because both somatic motor and somatic sensory are so important for survival, that's why those neurons are large and heavily myelinated. Group B fibers are lightly myelinated and are intermediate in diameter. So these are again somatic, these are autonomic nervous system neurons and interoceptors. Let me just go back. Let me fix that here real quick correctly. Okay, so type B fibers are going to be visceral motor and visceral sensory. Same thing with group C fibers, which are very small and unmyelinated. They will also be visceral motor and visceral sensory. Whereas group A fibers are going to be somatic motor and somatic sensory. Okay. So you should be able to distinguish between continuous propagation and saltatory propagation, and know that myelinated neurons can go through saltatory propagation, which will make these neurons very, very fast as far as the action potential will travel very quickly down these neurons, versus lightly myelinated or unmyelinated neurons in which the action potential will travel much slower. Okay, so this is the conclusion of PowerPoint B. I will go ahead and record PowerPoint C tomorrow.